Friday morning, June 28, 1991. We are on the first floor of the biomedical building on the campus of Brown University, talking with Dr. Boris Rotman about the history of the cell sorter. Dr. Rotman, you have kept a very rare prototype version of the cell sorter cell analyzer that was developed at Los Alamos. How did you acquire it, and how did it get to Brown?
I met Marvin Van Dilla through a letter he sent me in 1964 or 65. He was interested in our work with fluorescent cells because he wanted to use living organisms in the instrument he was building at Los Alamos. And so I visited him there, and I saw the instrument he had at the time, and obviously I realized the utility of such an instrument. But then it was not until I moved. At the time I was at the Stanford campus working for Syntex, Institute of Molecular Biology. And when I moved here to Brown, we started a collaboration with Marvin, and I was supplying cells, and so I became aware of the potential uses of the instrument, and I wanted one, and Marvin was nice enough to offer me to build a copy of it at Los Alamos. And he did, and sent it to me, and from the inventory numbers I can see that was in 67 when I received the instrument. And luckily I never tinker with it. I'm not an electronic expert, and I used to call Marvin every other day about little things on how to make the instrument work. So I kept the instrument as it was originally, and as far as I know, it's still in working order. The last time I used it for experiments was in 1981. My major work has been on doing viability studies for measuring antibody effects on cells, like killing antibodies, cytotoxic antibodies, and things like that.
Could you give us a brief tour of the instrument and maybe just point out some of its major parts and features?
The functional part, the most important part of the instrument is here. This is the chamber of the instrument where the cells flow. They are contained in this tube here, and they flow from underneath the chamber. Then is a water reservoir that feeds the outer sheath, as it's called, that goes around the cells and flows upwards. Here is a mercury arc lamp that illuminates the chamber, and one uses this microscope to see the intersection of the cell flow and the light, and uses this crude mechanism to center it. This is from, I believe, from an old microscope, was adapted for a micromanipulator, I'm not sure. And then here is the optical part. It's a lens and a photomultiplier that looks at the intersection of the beam. So I remember that when I first got the instrument, I had a really hard time. I couldn't get it work at all for a couple of weeks because, as you can see, it's very crude. This is a prototype. I don't know what vintage it is, whether it was the second prototype of Marvin or Mac. I'm not sure. But everything was movable. So until one obtained a signal on the oscilloscope, one was working absolutely blind, and I moved things up and down and couldn't get signals for a long time, and I would call Marvin, what's wrong? And but finally I got it to work, and once it's working and one doesn't touch anything, it works very well. So the rest of the instrument is mainly to detect the pulses that the photomultiplier sends out. So we have a preamplifier here. Then what is this could be described, was called at the time a 400 cell analyzer. It's a precursor of a computer. Nowadays, all of this probably could be done by a very simple computer. But at the time, one had to send the signals to the analyzer and sort them in the analyzer, and then show them in the oscilloscope. And also, I added a counter here to record the number of cells going by. I was more interested in the quantitative aspects of the instrument than in the sorting. The sorting part is in the back and over here. This is the most important part of the sorter, is the oscillator, but I've never worked with it, so this instrument has never been used really. Brand new. This is a plastic rod that oscillates, vibrates, driven by the instrument in the back, the oscillator here. And the liquid is fed into this oscillator, and the droplets come at the bottom of this. And here I should have had the separator plates to have the droplets go one way or another, but I never added that part because I discovered that my uses of the instrument for my research were more analytical than anything else.
The idea being that you wanted a single file of cells coming down, and the oscillator helped break up the stream of cells into individual droplets that then could be charged and later deflected and sorted.
Right. That's the principle of the cell sorter, where before one can send signals to the oscillator, one has to detect the cells. And the detector is what is most important. Even today, I would say most people who have a cell sorter use it mostly for analytical applications rather than separating cells. And so my instrument was used only for analytical.
Now you have your hand near the panel. Could you say something about the panel?
Oh, this was the artistic part of the instrument. This panel controls the flow of water into the cell, and as you can see, there are 5 inlets here, each one of these inlets go into a window. They are for the purpose of avoiding air bubbles in the chamber. And the art part became that you have to, when you got an air bubble that you saw under the microscope, then you start manipulating these things and try all kinds of things to get rid of that bubble. And some people were better than others to use the instrument. One of my technicians will come and say, can't get rid of the air bubble. So, oh, well, you have to go first on the central, wait a few seconds, turn it off, then go with the vacuum a little bit. And so this was the fun part of the instrument. The focusing was the most difficult thing. But then for day-to-day use, the operation of the water and the vacuum was essential.
Now, what is the photomultiplier and the lens resting on? It doesn't look like standard laboratory equipment.
No, this I think must have been an old lathe, an adaptation of a lathe. It has an up and down crude movement here in the back, and then has screws that allow you to move up and down and sideways. And as you can see, everything is very loose. But as a working model, it's a very useful instrument because it allows you to move everything. So I understand why this prototype was built in this way.
How much did it cost you to reproduce this?
Well, I paid $5,000 for the major hardware, not counting the electronics. So compared to the actual price of a sorter in the 100,000s, I think that I made a good buy.
Weren't you ever tempted to get the fancy commercial models when they started to be manufactured?
As a matter of fact, we did buy one Brown about 10 years ago. That is still in use. Nowadays, every department has a cell sorter or more. But for the cell sorter of Brown, I have to stand in line to use it. While for this one, I could do my experiments whenever it was convenient. So I kept on using mine other than the fancy one available in the division.
Maybe we'll break now. Start closing.
To caption.
Okay.
Yeah, so you sort of have to agree where you're going.
Give me one.
Okay. So let's have Boris identify the
Oscilloscope.
right to that.
Yeah, yeah.
You just hold on.
Okay.
If the screen is just here. Yeah, so you're gonna have to overshoot a little bit. Yeah. Okay. Roll.
Could you describe the oscilloscope and the electronic rack in a little more detail for us, Dr. Rotman?
Yes, this is the oscilloscope where one sees the outcome of the experiment. Usually it is composed of a couple of curves indicating the population of cells with certain fluorescence. The oscilloscope, the signal for the oscilloscope comes from these electronic components that are in essence way to sort out the signals. It's a 400 word analyzer was called. I don't know from where they came the word, but what this instrument does is to put in 400 pigeon boxes the pulses that come in. So if a pulse is, let's say, half a volt, it goes into a half a volt box. The next one is 0.7 volt, goes into another box. And at the end of about 1 or 2 minutes, then displays all the boxes here. So what you see here are 400 boxes and the number of pulses on each box. That is this. And then the bottom part of the instrument allows you to count each box, know exactly how many pulses one obtains in each box by this scaler. So we were able to do counting of viable cells in a very accurate way with this instrument and using much less cells. To give you an idea, in a normal laboratory, you need about 1 million cells per ml to count. Here, we could count with about 5,000 cells per ml, and much faster and without the subjectivity that the other counting has under the microscope. I am surprised that not many people have used a sorter for this type of work. It has been used more for fluorescent antibody work and not for quantitative cell counting.